Hey guys, in this video we're going to look a little bit more about DNA itself. So we talked about most of the time that we find blood at a crime scene or something like that, we initially just blood type it and don't actually look at the DNA itself. Whenever we have to dig a little bit further is whenever we're going to find more individualized evidence by using DNA fingerprinting. DNA fingerprinting is a common way to identify people by their unique genetic code. This is currently used during investigations to identify the perpetrator in a crime, to actually match the DNA to DNA at the crime scene. It's also used to identify fathers in paternity cases and to help identify remains in mass disasters and other situations where there are several bodies and often they cannot be identified. DNA can be found in every nucleated cell in the body, and DNA can be extracted from blood, semen, urine, bone, hair follicles, and saliva. So blood isn't the only thing that we can use to find that DNA match. So the basic functional and structural element of all living things is the cell, if you'll remember that from biology. And cells can function on their own, like red blood cells that are single-celled and function properly, or they'll function as a group in some of your larger tissues and organs. In the nucleus of all cells are our chromosomes, and chromosomes are long thread-like groups of genes found in the nucleus of a cell that contains DNA. And remember, your chromosomes and your DNA is inherited from both parents. So you will have traits from your mother and traits from your father. And DNA, or deoxyribonucleic acid, is the hereditary material of organisms. So that essentially makes us who we are. The human body is going to have approximately 35,000 genes. And a gene is a specific sequence of nucleotides in the DNA that codes for a specific body function. So this is what actually codes to make our proteins to allow our cells to have certain functions. And a protein is a component of all living cells, including enzymes, hormones, and antibodies. But remember, they're the ones that have the functional groups on them and give our cells a certain purpose or a certain function or job to do. Each gene is going to code for a specific body function or hereditary trait. This could include your hair color, eye color, facial features, so what type of nose, ears, eyes you have, things like that. It can also code for different diseases or disorders, depending on where it's located. And a specific gene can be carried on multiple chromosomes. So a lot of times you may have multiple places that code for hair color or multiple places that code for eye color, things like that. The DNA on chromosomes is made of 4 bases that are paired together and held with hydrogen bonds and a sugar phosphate backbone. So remember, DNA is that double helix that we learned in biology. On the side, you have that alternating sugar phosphate backbone, and then the bases are in the middle. Those are like the rungs of the ladder. So the bases are all considered nitrogenous bases because they all have a nitrogen backbone. The bases that pair together are adenine and thymine. So these are going to be connected with 2 hydrogen bonds. And guanine and cytosine, these are going to be connected with 3 hydrogen bonds. So remember, the A and the T go together, the G and the C go together. Each base is connected to a sugar molecule and a phosphate group. So whenever you have that nitrogenous base, the sugar, and the phosphate, that all together makes up a nucleotide unit. And the sugar in DNA is considered deoxyribose, which is where it gets its name from. The structure of DNA is important for proper function. If our DNA becomes damaged, our cell cannot function and reproduce properly. And DNA is unique in function because it can replicate itself. That's rather unique in comparison to other cells and stuff like that. DNA is arranged in a right-handed double helix, so it does twist to the right, but remember it's like a ladder where it has those two sides with the sugar and the phosphate. So that's the sides of the helix. And then going across are those nitrogenous bases connecting the two sides. The sides of the helix, the sugar and the phosphate group, is what gives DNA its acidic properties. So that's where it's the deoxyribonucleic acid. And remember the inside of the helix are those base pairs. And the average DNA molecule is going to have about 100 million of those nucleotide groups. In humans, the order of these nucleotides is going to be 99.9% the same. So all of us have 99.9% matching DNA. But what we tend to focus on is that 0.1% that's unique that makes each human different. The exception, of course, is if you have identical twins that will have identical DNA. If fraternal twins will still have different DNA even though they're born at the same time. The sequence of these nitrogenous bases is a code for amino acids. And amino acids are organic compounds with an amino group and a carboxylic acid. The amino acids are what are used to make your proteins. And your proteins, remember, all have certain functions. They're going to determine your growth, function, and reproduction in all of your cells. How DNA is used forensically. So blood and bodily fluids are some of the most common pieces of evidence that are used for testing DNA. But remember, it can also be used on skin cells and stuff like that as well. Remember, your blood is made up of 4 parts. It's made up of your red blood cells that carry oxygen, your plasma, that's the fluid that allows the cells to flow, your platelets that are used for clotting, and your white blood cells, which defend against infection. Those are the ones that produce the antibodies. One thing is your red blood cells do not have a nucleus and they do not contain DNA. That is one cell in your body that is non-nucleated. Whereas your white blood cells do contain DNA. So a lot of times whenever we're testing for DNA, we're testing the white cells, not the red cells. Excuse me. And a drop of blood can contain anywhere from 7,000 to 25,000 white blood cells of DNA. So even a very small sample will have a lot of these cells present. And only a few white blood cells are needed to extract DNA. And one reason that we can use such a small sample is because we have PCR or polymerase chain reaction. This we're going to talk about a little bit more later, but it's a lab technique used to make multiple copies of DNA for testing or characterization. So even if they have a single cell, they'll start with that one single cell of DNA and copy it to where we now have 2 copies. Those 2 will go through to make 4 copies. Those 4 copies will go through to make 8, and this will continue until we have a large enough sample to test. DNA fingerprinting or profiling can be useful for a lot of purposes. Like we said, it can be used to identify potential suspects whose DNA may match evidence left at crime scenes. It can clear persons wrongly accused of a crime. So remember, we don't always use evidence to say someone's guilty. We can exonerate them as well. We can identify crime and catastrophe victims, establish paternity and other family relationships, such as if they're siblings. Excuse me. And we can match organ donors with recipients in transplant programs. because remember you have to have a proper blood type match and everything, or else the organ will not function and it will die off. Samples collected at a crime scene are examined to figure out whether the sample is appropriate for DNA analysis. So they can't just test everything for DNA. So they really have to be able to prepare a sample in order to test it for DNA. So if DNA is on an object, so say it's on a shirt or a rug, it has to be removed from that object first. The DNA has to be extracted from the cell. So they actually have to get rid of all other cellular components other than the DNA. So they'll get rid of all the fats, proteins, and carbs. And once all those cellular components are removed, they'll use enzymes to release the DNA from the chromosomal packaging. So it will actually unwind the chromosomes. Um, and we said we use enzymes. Those are proteins that cause reactions to occur that's a rate sufficient to support life. And once the DNA is extracted, then it can be characterized. But it cannot be characterized whenever it's still in the nucleus. That's where we're going to stop today. In our next video, we're going to go over how we're going to characterize this DNA.